Dear learners, today we will focus on the topic of southern blotting. But before I jump to the topic, let us understand what is blotting in brief. What do you mean by blotting? A blot is a molecular biology and genetics is a method of transferring proteins, DNA or RNA onto a carrier. The term blotting refers to the transfer of biological samples from a gel to a membrane and their subsequent detection on the surface of the membrane is done. Technique for transferring the deoxyribonucleic acid, that is DNA, or ribonucleic acid, that is RNA, and proteins onto a carrier so that they can be separated, and often follows the use of a gel electrophoresis. This is the basic concept of blotting. There are three types of blotting technique, which is named as southern blotting, northern blotting, and western blotting. The southern blotting, it is used to detect the DNA. Northern blotting is used to detect RNA, and the western blotting is mainly used to detect the protein. What do you mean by southern blotting? Do you know why it is called as southern blotting? Because the method is named after its inventor, the British biologist Edwin Miller. In this particular slide, you can see one image of the British biologist. A southern blot is a method which is used in molecular biology for the detection of specific DNA sequence in DNA samples. Southern blotting combines transfer of electrophoresis, that can be separated DNA fragments to a filter membrane, and subsequent fragment detection by the probe hybridization. This method is named after its inventor, as I have already mentioned that. The principle of southern blotting says that the key to this method is hybridization. Let us know what is hybridization. Hybridization is a process of forming a double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target DNA. There are two important features of hybridization. Number one, the reactions are specific. The probes will only bind to targets with complementary sequence. Now, what is specific reaction? If the antigen specifically binds to the probes, that is called as a specific reaction. Number two, the probe can find one molecule of target in a mixture of millions of related but non-complementary molecules. Now let us focus on to the detailed procedure in performing a southern blotting. How to do a southern blotting? The various steps involved in southern blotting are: number 1, extract and purify DNA from the cells. Since DNA identification is done in this method, a DNA is need to be extracted and then purified. Number 2, the DNA is restricted with the enzymes in order to immobilize the activity of the fragment. Number 3, it is separated by the electrophoresis. Electrophoresis is a process in which the separation takes place on the basis of electric charge. Number 4, denaturation of the DNA, that is done to convert it to single strand. Then transfer to nitrocellulose paper. After that, step number 6 is addition of a labeled probe for hybridization to take place. Labeled probe is referred to a radio labeled probe. Next is washing off the unbound probe, and the last step is audio radiograph. This is the diagram of the different parts of an apparatus setup for the southern blotting. It consists of the weight, the paper towel stack, Whatman paper stack, the membrane, the gel which will be used for southern blotting, Whatman paper, saran wrap, the platform which will be supporting the whole setup, and the transfer of buffer is done at this position. Let us now discuss about the detailed procedure stepwise. Step number 1 is extraction and purification of DNA from the cells. In this step, the isolation of DNA in question from the rest of the cellular material in the nucleus is done. Incubating the specimen with detergent to promote the cell lysis is done. Lysis frees the cellular proteins of the DNA. Cell lysis occurs and the cellular proteins comes out from the DNA. Proteins are enzymatically degraded by incubation with a proteinase enzyme. Organic or non-inorganic extraction removes the proteins. DNA is then purified from the solution by alcohol precipitation procedure, and the visible DNA fibers are removed and suspended in the buffer. This step is followed by step number 2, that is DNA is restricted with the use of a restriction endonuclease enzyme as shown in the figure. Next step, that is step number 3, is separated by electrophoresis. The complex mixture of the fragments is subjected to gel electrophoresis in order to separate the fragments according to their size. Here in this diagram, you can see the setup of a gel electrophoresis tank. These are the wells where you put the sample and then the sample is allowed to run throughout the agarose gel in the presence of a visualizing agent, which can be any agent. A mutagenic agent is also there. Okay, so you can see that this is how you get to see the plates of the DNA sample. This is the agarose concentration plate which is observed under the PCR. Okay, next is denaturation of the DNA. How does denaturation occurs? Denaturation of the DNA is done by the restriction fragments that are present in the gel, and these are denatured with the help of alkali. This causes the double-stranded DNA to become single-stranded. DNA is then neutralized with the help of sodium chloride to prevent the rehybridization before adding the DNA probe into it. This is the setup which is used to carry out the denaturation of the DNA. Here you can see the salt solution is there. This is the filter. These are the paper towels. Here you place the gel and this is the sponge where you put the sample. Step number 5 is to transfer to nitrocellulose paper. In this step, we are transferring the DNA from gel to a solid support, that is blotting. This blot is made permanent by drying it at around 80 degrees centigrade. And also it can be achieved by exposing to UV irradiation. Step number 6 is adding a labeled probe for hybridization. In this step, the filter is incubated under hybridization conditions with a specific radio-labeled DNA probe. Labeled probe means it has to be radio-labeled. The probe hybridizes to the complementary DNA restriction fragment. Next, about step number 7. In step number 7, the washing off of the unbound probe is done. Once it is allowed to bound, the bounding takes place at the previous step, that is step number 6. After that, this is washed with the buffers which contains sodium chloride and detergent to wash away the excess probe and reduce the background. So now it only contains the probe, DNA probe, which are bounded. The last step is autoradiograph. If the probe is radioactive, since it is a labeled probe, radio-labeled probe, the particles emits when exposed to X-ray film. There will be dark spots on the film wherever the probe is bound. So here you can see that the probe is hybridized to complementary sequence, and this removes the unbound probe of the washing. The probe is now hybridized. Now this is exposed to the X-ray film to filter, and this is what you see as a result. The dark bands represent the bound probe. This is the whole flowchart form procedure of the southern blotting. Okay, here you can see all the steps are mentioned with the help of a picture. First of all, the RNA and the DNA is taken on the plate, 32P labeled size markers. Electrophoresis is done. Migration is done. The solution is run. Agarose plating is done for gel electrophoresis. Then the agarose gel is observed under the PCR to check the presence of the bands, DNA bands. Then you use this particular solution to pass through the gel. The paper towel is there. You place the band on the nitrocellulose filter. From there, the filtration, the DNA is transferred to the filter and allowed to hybridize with unique 32P labeled nucleic acid probe. Then washing off is done so that the unbound probe are washed away. The bound probe is hybridized to complementary sequence and then it is exposed to the X-ray film. The dark band shows the bound probe, that is the autoradiogram. In this particular slide, we will study about the various advantages and disadvantages associated with this technique. If we focus on the advantages, we will get to know that it is an effective way to detect a specific DNA sequence in a large complex sample of DNA. It can be used to quantify the amount of the present DNA, that is, quantitative analysis can be done, and is definitely a cheaper method than that of the DNA sequencing. Now we'll study about the different disadvantages. It is more expensive than most of the other tests. It is a complex and labor intensive method and it's quite time consuming and cumbersome. There's a wide field of application for southern blotting. Let's have a look on what are they. It is applicable to identify specific DNA in a DNA sample. It is also used to isolate the desired DNA for construction of rDNA, to identify the mutations, deletions and DNA gene rearrangements. It is used in prognosis of cancer and in prenatal diagnosis of genetic diseases. It is used in RFLP. It is used in the diagnosis of HIV-1 and infectious disease. In DNA fingerprinting, it has an application in paternity and maternity testing, in criminal identification and forensics, and personal identification. So you can see it has a wide range of application. We conclude the lecture. Hope this presentation has helped you to understand the southern blotting technique, its underlying principle, process protocol, and field of application. If you have any queries or suggestions, feel free to mail me at the given mail ID. Thank you for listening to the video and following my lectures.